So hi everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Elizabeth Mormino. Dr. Mormino completed a PhD in neuroscience at UC Berkeley in the laboratory of Dr. William Jesus, where she performed some of the initial studies applying amyloid PET with the tracer PIB to clinically normal older individuals. That was in order to provide evidence that early pathophysiological processes of Alzheimer's disease are associated with subtle changes to brain regions critical for memory. During her postdoc fellowship at MGH with Dr. Risa Sperling and Keith Johnson, she investigated longitudinal cognitive decline in preclinical Alzheimer's disease. In 2017, Dr. Mormino joined the faculty at Stanford University in the Department of Neurology and Neurological Sciences, where she leads the imaging core of the Stanford ADRC and co-directs the Stanford Aging and Memory Study. I would just like to remind the audience to please address any question you have using the Q&A box or raise your hand. Dr. Mormino, thank you very much for coming here today. The virtual stage is all yours.
Thank you. Thanks for the invitation. It's really nice to be here. So I did my postdoc at the Martinos Center, and I really miss. There's so many aspects I miss of the Martinos Center. I definitely miss all the people I used to work with. I miss the salad bar. I don't know if the salad bar is still there, if it's going to survive COVID. But there's so many, and I definitely miss the Navy Yard in general, being a walk along the water. So yeah, thanks for having me. It was really nice to get this invitation for me. If there's questions in the middle, I'm okay with fielding them. I know it's a little kind of awkward with Zoom, but I'm gonna try to have the chat box up. So if you do have a question that makes sense to ask in real time, feel free to just enter the question and I'll do my best to pay attention to that. Okay, let me move a couple things on my screen. All right, so for my talk, here's an overview of what I'd like to cover. So I'm going to start with a brief background on essentially the concept of preclinical AD, and I just want to take some time to highlight a couple kind of big picture patterns that have emerged in the space with respect to early amyloid and tau in normal aging human cohorts. This is a rapidly evolving field, and many of the folks at the Martinos Center and MGH are leaders in this space. So I think this will be quite familiar to most of you, but there's also been a ton of work in this space as well over the last couple of years, especially with respect to tau PET, and I think there are some kind of patterns that have emerged that are quite important. So I'm going to touch on those. And then I'd like to introduce some local data collection efforts we have at Stanford, the Stanford Aging and Memory Study, and present some of our work from that effort. And then I also want to present some tau PET data from publicly available data sets that we've been working on quite consistently this last year or so. Both from the A4 study and also from ADNI. So as you guys know, the beta amyloid plaques and the neurofibrillary tangles are hallmark pathological features of Alzheimer's disease, dementia. And even though we know that they're central for this disease, they're also common among normal older individuals that have no clinical symptoms whatsoever of dementia or even mild cognitive impairment. This group has been of great interest over the last 15 plus years and is broadly referred to as preclinical AD. So this idea of a time in which the pathophysiological processes of the disease have begun, but there's no cognitive consequence just yet, or at least nothing that's clinically meaningful that would warrant a diagnosis of MCI or dementia. And during this stage, so we tend to see amyloid plaques in these folks. If we look at the prevalence of this as a function of age, we'll find that about 30% or so of those in their 70s, or about 25% or so of those that are about 70 with no symptoms whatsoever in this normal control group will show what we refer to as amyloid positivity. The signal, as we know from the postmortem literature and from imaging, is quite distributed even early on. It tends to affect most of association cortex. And we also expect there to be some degree of neurofibrillary tangles. So tangles, unlike plaques, have a much more focal pattern. So they tend to begin, at least from the postmortem literature, we know they tend to begin from the entorhinal cortex before spreading to other limbic regions, and then eventually they impact other cortical regions as well. And within normal controls, what we would expect is that this entorhinal tau is beginning, quite early, such that amongst older individuals, there should be some tangles, in the entorhinal cortex. This is thought to occur before this amyloid positivity stage. And then somewhere after or during the amyloid, this amyloid stage, we might start to see more neocortical tau. But the main point, like for thinking about preclinical AD, is that we do expect, both this abnormal amyloid signal to emerge in some individuals, and then also some more focal tau patterns as well. And as you guys know, we can now measure these pathologies using PET, and a tremendous amount of work has been done in this space over the last 15 years or so. Here's just 3 example cases. So here's an amyloid, what we would call an amyloid negative normal control. Here's their amyloid scan, and here's their corresponding tau PET scan. You can see this kind of pattern of white matter uptake, which we just consider that to be off-target binding. That's not actually binding to the plaque itself, but it's very typical of these ligands. And here's their corresponding tau PET scan. I mean, there's a little bit of signal here and there, but that's most likely actually some choroid plexus off-target binding. Here's an amyloid positive normal control. So about the same age as this person, but here you can see a very different pattern with respect to their amyloid signal. There's this warm uptake throughout cortex here. So this is a very obviously amyloid positive scan. And interestingly, here's their corresponding tau PET scan, and you can see this nice signal in the medial temporal lobe that extends into the lateral temporal cortex in this case. And then just for comparison, here's an actual amyloid positive patient with Alzheimer's disease dementia. And you can see for both modalities, there's pretty widespread involvement for both amyloid and tau. And as I just mentioned in the last slide, so this is data from the Harvard Aging Brain Study. If you were to scan, a number of older normal controls with amyloid PET, we pretty consistently find that about, depending on the age of the older cohort, about 30% or so will end up having this kind of elevated signal that's on par with the AD dementia cases. And now more recently with tau PET, we've been able to dig into what the actual distributions of this signal looks like in normal aging cohorts. So I just want to review kind of a couple points. So one, this is a study from the Mayo Clinic Study of Aging, and this is just simply showing, in this case, entorhinal cortex values on the y-axis, and then age on the x-axis. And these are all clinically normal individuals. The black dots are just their younger subjects under age 50. And then the blue diamonds are their amyloid negative group, and then the red circles are the amyloid positive group. And what you can see is that, for one, there is this pretty dramatic effect of amyloid positivity. So we don't really see elevated values in tau PET until you're amyloid positive. So that's shown with these red guys. And really, it's not that many cases. So these very high cases are very rare and uncommon amongst normal control cohorts. So when we're looking at tau PET signal, we're really examining a restricted range of values. You can also appreciate a slight, age association between this signal, but that is embedded within this larger effect related to amyloid status. So that's one kind of point about the tau PET side. And another point, which I'm going to come back to at the end of the talk, is the regional pattern of this elevation. So here I'm showing voxel-wise maps across 3 independent cohorts. So this is work that Keith Johnson and Aaron Schultz did a few years ago now. This is from the HABS study, and this is essentially just showing all the regions where there's elevated tau PET signal in amyloid in individuals with high amyloid compared to low amyloid. And what we can see is involvement, as expected, of the entorhinal cortex and medial temporal lobe, and also some pretty dramatic involvement of ventral temporal cortex. So this whole kind of ventral side of the temporal lobe kind of bleeding into the lateral side, as seen here. This is an analogous map for the A4 data set, which I'm going to present some data, some of our work on this at the end of the talk. But here we see the medial temporal lobe involvement, the ventral temporal cortex involvement, just like in HABS, that bleeds into lateral temporal. But also here we start seeing angular gyrus and also precuneus as well, suggesting that there are more kind of cortical hubs that could be involved with this early tau PET signal. And then finally, here's data from the Wisconsin group with a similar contrast comparing amyloid positive normals to amyloid negative normals, and a very similar pattern has emerged in this cohort as well, even though they're using a different tau ligand. Again, entorhinal cortex and the medial temporal lobe, they don't have a ventral view in their paper, but you can kind of see the ventral kind of side bleeding into lateral temporal, and also this same kind of inferior parietal angular area and the precuneus. So this is quite interesting. I mean, I think, I'm going to show a lot of data kind of centered around the concept of the medial temporal lobe, but I in particular have become very interested in these initial kind of cortical spots of tau elevations that tend to be present in these normal control cohorts. So in addition to those maps, which are all centered around the contrast of amyloid positive and negative, there is also work reminding us that there is this early signal that is likely more focused in the entorhinal cortex that might be more age dependent rather than amyloid dependent, or have at least both components. So this is work from the Biofinder group in Sweden, highlighting this early kind of this early stage within entorhinal cortex, and also showing that this tau signal in entorhinal cortex also corresponds with structural measures in the entorhinal cortex, an effect that persists after controlling for amyloid, so may not be amyloid dependent. And here's beautiful work from Keith Johnson's group, also highlighting this ability to detect this very early focal tau near the rhinal cortex, and also showing that it has this very nice age effect in the amyloid negative groups, in addition to this effect of amyloid positivity. So here's the amyloid positive group, do definitely have this elevated rhinal signal with tau PET, but also, it is striking that there's on top of that, within this an age effect as well. So it is exciting that tau PET, in addition to finding this kind of amyloid positive signal that may be more robust, may also be able to pick up this focal signal that is age related, that probably precedes overt amyloid positivity. And then finally, I think this is my last background slide. So one finding that has also been quite consistent in the literature thus far is that elevated tau PET signal within normal aging cohorts seems to be associated with lower episodic memory performance. So here's scatter plots, kind of highlighting the amount of data that has been collected to get at this question, and also highlighting some consistencies between HABS, this Wisconsin study that I mentioned earlier. This is work from Berkeley, and this is work from Mayo, but all essentially find that if you have higher tau PET signal, and most of these studies are looking within the entorhinal cortex. So if you have this higher tau signal, you tend to have lower memory. The main difference in inconsistencies in the literature is whether or not this is amyloid dependent. So for these two studies, for HABS and Wisconsin, the effect of elevated tau is really driven by the folks that have high tau and also are high amyloid. So that in HABS is shown by these red diamonds. They're amyloid positive. They tend to have higher tau, and those are the folks that have also subtle decreases in memory. And it's the same situation for RAP. For Bax and Mayo, they actually find that it's independent of amyloid. So here, the red dots are also the amyloid positives. They tend to see more of a continuum. So they tend to have these high tau cases that are not amyloid positive. So actually their highest tau case is an amyloid negative. This data from Mayo is not color-coded by amyloid status, but in that paper, they do look at the amyloid positive and negative groups separately and do find that there is a relationship with memory independent of amyloid status. So there are still some, there are inconsistencies with that finding, but I would say that overall, it seems like if you have elevated tau and you're a normal control, that tends to be associated with this subtle decreases in memory as well. Okay, so that was just meant to be just kind of a brief orientation and background on kind of preclinical AD and how we can use these tools to measure these early changes in those cohorts. Now I'm gonna switch gears and talk about the local study that I'm involved with at Stanford, the Stanford Aging and Memory Study. So this slide is meant to give you kind of a brief background of the study and also what we have now and where we're hoping to go. So this study, it was initiated in 2014 by Anthony Wagner, and it was designed to be kind of a multimodal imaging baseline study of aging, and it's completed. So all the data for this has been collected, and that was recently finished about just right before COVID, actually. And as part of this baseline study, each individual came in, these are all older subjects above age 60, for a 3-day visit, and they did a whole, a very intensive kind of battery of imaging and other visits. So they did a lumbar puncture, to collect CSF to assess AD markers, amyloid and tau. They did a 3T functional MRI scan of associative memory, both during the encoding phase and also during the retrieval phase. I'm going to be presenting some of that data in the next few slides. They also underwent a 7T structural MRI scan to be able to get at subfields within the medial temporal lobe. And the overall idea, was to understand, what are the drivers of individual differences in memory, in particular associative memory, and how do these processes during successful memory relate to changes in structure and also to early AD. So it's a really exciting baseline data set, and we're still sorting through some of the analyses. The downside of having such a big, large study spanning 5 years of data collection is, as you guys know, everybody's imagers here, it is pretty complicated with all the different software updates and the analysis streams, to process all this data, but we're hoping it will all be, done somewhat soon. But the data is in hand, which is really exciting. And then, so I joined Stanford in 2017, and Anthony and I started kind of collaborating on some ideas on how to further enrich kind of this baseline data set. And so 2 things I was interested in was integrating tau PET. So I wrote an R21 to integrate tau PET into this cohort. So I was able to capture some of the remaining subjects as they were going through their baseline visit, and collected about 40 tau PET scans on these individuals. So that obviously, gives us further phenotyping, especially with the medial temporal lobe focus of this study. It allows us to look at tau PET within the medial temporal lobe. We've used PI 2620, so that's been an interesting experience. It's one of the newer ligands. And then I also was interested in extending this into a longitudinal study. So it is, really interesting to be able to relate these things concurrently, but a lot of the power in these normal aging cohorts is the ability to look and see what happens over time and how these sort of baseline measures predict change over time. So we wrote some pilot funding to extend, or essentially to show feasibility that we would be able to invite these people back for a second visit. So we did that for about 50 subjects or so, to correspond with their 3-year post-baseline visit. And there we repeated the 7T and the lumbar puncture, and also had tau PET in that study. And that was, it was good. It was a lot of work, but unfortunately with these normal cohorts, 50 subjects isn't really enough. You really need to have a larger cohort to be able to detect these changes. So I right now have a R01 that's pending. It's in this resubmission stage to extend this whole cohort into the longitudinal phase to be able to try to bring back as many folks as possible to be able to determine, what are the set of predictors most predictive of change, both in the imaging metrics and also memory. So we'll see what happens. Fingers crossed. It's kind of out of my hands at this point, but I would really love to be able to follow these folks over time. So as I mentioned, this cohort is it's a little over 200 subjects at baseline that have undergone most of the imaging. It's only, so for CSF, we have 150 that completed the CSF. So not everybody did CSF. We had some, some people decided not to do it. And then also, there's some people we couldn't do it with the pre-screens, but the majority of subjects do have CSF, and we were able to scan most of the people missing CSF with amyloid PET to be able to have some sort of amyloid status on these folks. The mean age is just shy of 70. We do have a higher proportion of females, which tends to be kind of common for these volunteering studies. The education is quite high. I mean, this is a problem for all these sorts of studies and is a major limitation. And also the breakdown by racial groups is also highly skewed towards white individuals, which is also a problem for generalizability. But overall, this is, a large cohort of 200, people that were clinically normal at baseline, characterized both with imaging and CSF markers. So I think, yes, forgot about this slide. So this is showing us just the distributions of the CSF markers that we have in SAMs at baseline. So this is as expected. So this is what the amyloid measurement looks like. We're using, so we had the benefit of being able to collaborate with our Alzheimer's Center's biomarker core, which has a new automated Lumipulse system in-house. So we were able to do a single batch analysis with this newer methods, which these analyses for CSF can have a lot of noise introduced with the more manual methods. So we were able to use this more updated measure, which also gave us the amyloid 40, which some people have argued, is a better way of treating this amyloid signal by normalizing for overall protein production. So that's the amyloid measure that we're using, is this ratio between 42 and 40. This is what the distribution looks like across the 150, and this is exactly consistent with other cohorts. We tend to see these two modes, with the smaller mode with lower ratios indicating more abnormal amyloids. This would be considered our amyloid positive group. And then this other mode reflecting the amyloid negative group. And this is just a simple bivariate relationship showing the association between p-tau, so this global measure of, presumably reflecting tau burden in the brain, with this amyloid ratio. And again, this is as expected. We see this inverse relationship because the lower ratios are more abnormal, such that if you're in this amyloid positive group, you tend to have higher tau levels. But again, there's a lot of variance, which is very similar to the tau PET slide I showed a couple slides back, that there's an effect of amyloid, but there's a lot of variance. There's some folks that are on the higher end, but many amyloid positives that have low levels of the tau measure. And I only have one slide on the 7T data, because this is something that Tammy Tran, who's a postdoc leading these analyses, she's still working through all the delineations. We're doing everything manually. But here's our preliminary glimpse at this data. We've looked at, we're delineating numerous regions within the hippocampus and also the entorhinal cortex. Here are the two regions that most of our hypotheses are centered around. So this is lateral entorhinal volume and also a measure of CA1, so the actual SRLM area right here of CA1. And so this is just simply showing the relationship between volumes and our p-tau measure. And so we don't see any relationships with lateral entorhinal cortex, but we do see this nice relationship with the CA1 measure, suggesting that these normal controls with higher global levels of p-tau are showing thinning, in this structure, which is in line with, with, as we know, CA1 being this initial area of tangle deposition during this early phase of tangle development. The lack of relationship with entorhinal, I mean, I've kind of interpreted that to suggest that this p-tau measure, this global measure of p-tau in the CSF is not going to be as sensitive to that very focal, initial effect, but it could certainly be an issue on the MRI side as well. And now I'm gonna switch over to our fMRI data from this study. So with the overall design, we were very interested in the regions that are involved with specifically episodic memory retrieval, which are highlighted in here from these review articles, and in particular for visual associative memory, which does, highly depends on activation both during encoding and retrieval in the ventral temporal cortex. The angular gyrus is probably involved for more for both stimuli dependent and also independent memory processes during retrieval, and of course the medial temporal lobe. But as I pointed out to you guys a few slides ago, it's so interesting in that these regions that are critical for memory retrieval also line up so well with the regions that we see, especially in cortex, for tau PET in normal controls. So again, I mentioned this earlier, but the VTC in particular seems to be a very robust early signal for tau PET, and then even the angular gyrus as well. So the study wasn't designed with that in mind. This was designed, before we were able to glean these insights from the tau PET space, but it is pretty striking the degree of spatial overlap between memory retrieval processes and early tau PET signal in normal older cohorts. So here's the task. This is an associative memory task where there's famous faces and places that are paired with unrelated words. In the scanner, we do this encoding phase where they're exposed to the pairs, and then there's also the test phase where they're given the word cue and asked what the corresponding visual stimulus is, and this is all done in the scanner, which allows us to look at in particular cortical reinstatement, which I'm going to describe in the next slide. We're very interested in the concept of activation during this phase and how it matches the encoding activation in VTC and angular. So this task was done in the majority of the individuals, and we do see, so here's just a simple relationship between the ability to perform this task, so to remember the associative pairs as a function of age. This is similar to what we'd expect. And from here, this is a very rich data set, and there's a lot of important questions that could be asked. So one, with respect to these retrieval processes, we're interested in particular cortical reinstatement. We do want to know if we can capture that within VTC and angular and understand whether that's important for actual memory performance. So both memory during the task itself and also overall memory. And of course, we are interested in whether or not this is related to early AD. And so here's kind of a schematic, showing the main measures that we've been focused on in a very a priori way. And most of this has been published without the AD piece just yet by Ali Trell and eLife. But essentially, we're interested in these retrieval markers. So what is the hippocampus doing when it's given this cue in terms of its activation? And then this concept of cortical reinstatement. So we're very interested in this because we know it's hippocampal dependent. As I mentioned, for visual associative memory, it depends on visual temporal cortex, given the visual aspect of the task, and then also angular gyrus. And here we're effectively, what we do is we train a classifier during the encoding trials, and we do this at the level of category. So we have a classifier looking at the places and understanding the activation pattern for each individual during that category, and then do the same for faces. And then we simply ask, how well is that, how much of a match is there during the retrieval trial of those categories compared to encoding. So that's how we do this kind of pattern similarity matching between those two phases, the encoding and the retrieval phase, to capture this cortical reinstatement, this idea that these patterns of activation during successful retrieval will match what happened during encoding. And so here we can ask, both about this kind of cortical process and also hippocampal activation during retrieval. How does this impact overall memory performance? And what we do see for both the hippocampus and also the reinstatement strength described here across both regions. So here BTC is shown in blue, angular is shown in this orange color. In both cases, the higher the strength of reinstatement, so the higher encoding, the higher that retrieval matches the encoding activation, the more likely that those trials will be an actual hit, something that will be remembered, is an item that's remembered, and the same for hippocampus activation. So both these metrics are telling us something about trial level differences in performance. And further, so in addition to the trial level performance, whether the trial during retrieval actually is associated with the successful memory of the visual item, we also can compute this overall measure of associative memory with this D prime measure, and also an average estimate of hippocampus activation, as well as these reinstatement measures from VTC and angular. And again, we see that this activation and also the reinstatement from both these regions is positively associated with overall memory on the task as well. And then finally, so this is our first attempt to integrate the AD biomarker piece into this. So first, so this is just the showing performance on this task, the same associative D prime measure I showed in the previous slide. This is just a function of age, which I already showed you that at the beginning. And here's this performance as a function of this amyloid measure and this tau measure. And we can see there's the expected relationships with both of these metrics. If you put both in the model at the same time, because they are related to each other, amyloid and p-tau. P-tau is the actual driver of this relationship as expected. So those with higher p-tau do perform worse on this task. And then finally, if we compare the CSF metrics directly to the fMRI metrics, we do find, a similar pattern as expected. So we see that for both hippocampus activation, if you have higher p-tau, you have less activation, your cortical reinstatement strength is reduced in both VTC and angular. And here's just a little glimpse of our PET data. So I mentioned, we do have PET data. It's on a very small subset, so it's a little embarrassing to show these sort of scatter plots that are so sparse. But we do see that it goes in the consistent direction as we see with the larger CSF sample. So overall, this is, I think, quite interesting. Showing this direct relationship between these key fMRI metrics, tapping into different sub-processes, critical for memory formation, and their relationship to this p-tau measure in normal controls. So, I mean, there's so many questions with this. So, and we'll be able to address this once we get a larger tau PET data set. But one question, especially with the cortical reinstatement measures, is whether or not this is actually mediated by co-local tau within VTC and angular. I was talking about this pattern earlier, that those seem to be very key early regions in terms of the cortical deposition of tau, or it could be related to distal medial temporal lobe tau signal as well. So we're hoping to flush this out once we get the tau PET data. And then also another question, I mean, all these things seem to be kind of, related to each other and all going, kind of painting this story of preclinical AD, where if you're kind of have these poor functional metrics and higher p-tau, then you'll have less memory. But once we have the full data set, we will be able to get into questions of mediation or independent prediction of actual memory performance as well. So the last, I have the last section, I'm gonna now shift gears to kind of focus even more on this concept of BTC and angular, tau PET, that I'm hoping to link back to the fMRI data in the next couple of years, but to try to further understand this signal in the context of normal aging. So I showed you guys these maps, highlighting from HABs and A4, this medial temporal lobe, BTC, and angular. Here's some work from Christina Young in my group looking at some broad patterns in the ADNI data set. So this first row here is showing cross-sectional associations with baseline tau in amyloid negative normals shown in pink, amyloid positive normals, and then amyloid positive MCI is kind of a comparison. And here we're plotting entorhinal, inferior temporal, which is one component of BTC, inferior parietal, which is the free surfer region most analogous to angular gyrus, and then middle frontal gyrus, and then also precentral. And so for a baseline, what you can see is, which is consistent with these maps, is that if you're amyloid positive, you do show elevations in the entorhinal, also in inferior temporal, and also in inferior parietal. So this is another cohort, kind of replicating these effects in these other cohorts. And then you don't see any difference in the frontal and precentral is kind of a control region. This bottom row is now showing the longitudinal data. So this is a longitudinal tau PET data. It's very short. It's only on average 1.5 years or so follow-up. So these are really measuring very short-term changes in tau PET signal. And what you see here is you don't see too many group differences with entorhinal. However, you do see that both normal control groups are significantly different than zero. So they're both accumulating tau and entorhinal. For inferior temporal, we do tend to see that the amyloid positive normals are accumulating more. But again, actually both normal groups are significantly accumulating. Both groups are significantly different than zero. That's what these pound signs are indicated. And the same for inferior parietal. So there is this difference in the amyloid positive group, and the amyloid positive group is accumulating over time. In this case, the amyloid negative group is not. And then we see no associations with these later regions with the frontal gyrus and precentral. And what's interesting to me about this is that we've looked at this very comprehensively across all the regions generated in the ASEG and APARC through the free surfer standard stream. And it's really just these three regions that show these significant effects, both longitudinally and cross-sectionally in the normal control. So it actually seems to be that these are quite specific regions that are showing this early signal, and it seems to also be consistent across now at least four cohorts. And then we started, thinking about these patterns, and I started wondering, we tend to think of tau as going through this sequence. So we tend to think of it, in kind of a Brock staging sequence where we start with entorhinal, then we go to hippocampus with CA1, and then other regions of the hippocampus. And then from there, maybe it would spread to BTC, maybe angular at the same time, or maybe it would be more linear like BTC and then angular. However, there's a lot of work in the more patient space, showing that there's actually different subtypes of tau deposition and patterns and atrophy that are associated with slightly different or with different clinical presentations as well. So another possibility rather than a sequence is like there could be subtypes. There could be an entorhinal kind of hippocampus medial temporal lobe path. And there also could be a separate path that goes from BTC or angular, or maybe, separately BTC and angular. So we started thinking about, could we actually try to get at this with normal cohorts? And as I mentioned, this concept of subtypes is very common in the patient space. So in AD, we do have these atypical presentations that will be very different than an amnestic presentation, for instance. So we have things like posterior cortical atrophy, where the atrophy and the tau pattern is very much an occipital, kind of posterior parietal, posterior temporal pattern. And those patients, they present with some pretty severe visual spatial deficits as opposed to memory. And that's just one extreme example. Brad Dickerson has published, consistently with dysexecutive, forms of AD that tend to have more frontal patterns of atrophy. So this concept of subtypes is very common in patients, but the idea of it in the preclinical AD space hasn't really been explored. This is a recent paper from the BioFinder group showing subtypes for tau PET in the context of patients. They did have normal controls, but the normal controls tended to be classified as tau negative using their algorithm. But this is just one example, which is consistent with this broader literature of these different subtypes. And what they reported is that one subtype would be this limbic subtype that is more medial temporal lobe. Another subtype is what was phrased medial temporal lobe sparing. So the medial temporal lobe is not involved as much, but different areas in cortex. And then a posterior subtype, which feels very much like the posterior cortical atrophy phenotype I just described, where it's much more occipital and posterior, the involvement. And then they also identified a lateral temporal predominant subtype as well, using tau PET data mostly from patients. So we were fortunate that the A4 data set, which you guys are likely very familiar with, it's designed to be a clinical trial of testing anti-amyloid therapies in the context of preclinical AD, but they have released their full kind of screening baseline data set for the community to probe. And what's really amazing about this data set, so they just released the tau PET data a couple weeks ago. I think one of the largest data sets of normal controls that are amyloid positive. So usually with a normal cohort, you would only have 30% are amyloid positive, and then within the 30%, there's some variance in the tau. So A4, it's kind of the opposite, where by the study design, they were recruiting specifically amyloid positives, and the amyloid positives were the ones that got the tau PET. So with this data set, there's nearly 400 amyloid positive normal controls. So that's a fair amount of variance to be able to probe questions like heterogeneity and the spatial patterns. And then they also have a smaller set of amyloid negatives for comparison in their group. So this is our first pass of trying to get at this concept of heterogeneity, really leveraging the size of this tremendous resource. And so our first passes work, again, from Christina Young and my group, has just been to do some hierarchical clustering of this data. We've just pretty much fed in the majority of free surfer. So we process the MRI with free surfer and then just extract the PET values from those different regions, and then put those regions averaged across hemisphere into this clustering algorithm. The regions are just organized by anatomy over here. And what's interesting from this, I was quite surprised, this is just the amyloid positives, so we didn't put the amyloid negatives in here, is that we do tend to see these kind of two medial temporal lobe clusters here that have different involvement. So the medial temporal lobe ROIs are kind of at the top here. So you can see the red means kind of greater contribution to the clustering. So here's a more restricted medial temporal lobe-ish subtype. This is a medial temporal lobe subtype that also has many of these ventral the ventral temporal cortex regions. And then these two clusters tend to be a little less consistent with their involvement in the medial temporal lobe, but tend to have more of this cortical presentation. So here's these VTC regions. And then this cluster it's a little less clear, but definitely has this inferior parietal, which is angular and also precuneus, and a little bit of frontal contributions as well. And just to stress this, so the medial temporal lobe, I kind of pointed out, so it seems like two clusters that are more on the medial temporal lobe side. VTC seems to be one of the most kind of robust contributors and differs across the subtypes. And then, angular keeps on coming up. Angular does seem to be this kind of core contributor to these two kind of non-medial temporal lobe subtypes. So I thought that was quite exciting. Here's how the actual individual data points fall to see. So one of the issues with these clustering algorithms is how much is it just kind of capitalizing on correlated noise. So here we're plotting the actual SUVR values for a couple key regions across the 4 subtypes. So here's the So this was that first kind of medial temporal lobe limbic guy. This is the limbic plus VTC. And here are the two cortical subtypes. And then in comparison is the amyloid negative group. So for entorhinal, we do see the medial temporal lobe clusters, do have higher entorhinal, and the cortical guys do not. And it's interesting, like these guys, the medial temporal lobe, they are significantly greater than the amyloid negative group, but these guys are not. Then for inferior temporal, we do see this, this one cluster that has medial temporal and VTC is elevated, but also this cortical guy is elevated. So this is not, elevated compared to the amyloid negatives. For inferior parietal, both the two kind of cortical predominant clusters tend to be elevated compared to the other clusters. And then for superior frontal, it's really just this last cluster that tended to have, a couple of kind of parietal contributors and frontal contributors. So importantly, all these different subtypes, do show elevations compared to the amyloid negative group. Again, suggesting that the algorithm is not just capitalizing on correlated noise. Another, so we are trying to follow this up to kind of validate these clusters and better understand these clusters, such as looking at relationships with cognition. But one thing that is also coming out just in simple demographics is that these cortical expressing clusters, the ones that tend to have less involvement of the medial temporal lobe, also tend to be younger in age. So that's also something that mirrors the AD patient space, since it's been shown that these earlier onset AD cases tend to have a more cortical representation compared to the medial temporal lobe. So this is quite interesting because it mirrors that, but this is in the context of an older aging cohort. We tend to think of those atypical presentations as being so uncommon, but here it seems like it's actually embedded within this, this aging space. Okay, so I will summarize, and then if there's questions, I'm happy to take some. So I showed you that both amyloid and tau PET, show early signal in aging cohorts, and they also have a specific relationship to each other, and they also are associated with changes in memory. It probably is going to be the case that that tau PET and elevations in tau, whether it be p-tau or regional tau, is more proximal to any sort of change in memory that we might detect. Even though we are finding these broad associations between tau signal and memory change, I think it's still the case that the actual mechanisms underlying these consequences of tau is unclear. And I think our work, both in the fMRI space and also more recently looking at these spatial patterns, is aligning to suggest that the consequences might not just be solely medial temporal lobe focused. It might be a blend of both the medial temporal lobe, but also these cortical processes. And tasks that tap into both of these potential pathways, may be particularly sensitive for capturing, both medial temporal lobe and cortical processes. Visual associative memory, given its reliance on both the medial temporal lobe, but also VTC and angular in particular, might be one example of a task that capitalizes for these early signatures. I think this recent work with the very large A4 tau PET data set does bring up the possibility that this early signal in normal aging cohorts is going to be more complicated than just Braak staging. So it seems like there could be different subtypes and heterogeneity, in these aging cohorts, that it won't be as simple as everybody has high entorhinal tau first, and that instead it could be the case there's different drivers of this. So age might be a primary driver of the entorhinal piece, whereas genetics or other exposures might drive these cortical patterns. I also think this heterogeneity concept is very exciting with the parallel to what we already know in actual patients, an idea of this being present, in the context of aging and preclinical AD. And I think this, importantly, does all have important implications for how we think about characterizing preclinical AD, our ability to measure the earliest deficits associated with these pathological processes, and also for how we think of tau PET in clinical trials too. Going back to this heterogeneity concept, if there is differences in spatial patterns, we might not be able to just use a simple, early medial temporal lobe ROI for assessing treatment effects and the association between tau and other outcomes as well. So that's all I have to show you. I'm just going to quickly acknowledge my collaborators at Stanford, in particular, Anthony Wagner and Brian Rett, who we've been working on this SAMS data together for the last few years and have been adding to that cohort and hope to continue to add to it. Within the lab, I highlighted the work of Christina Young, who's a postdoc working on the tau PET data, Tammy Tran, who's working on who leads the 7T data, and then Ali Trell, who works with the fMRI data from SAMS. And I, of course, want to acknowledge my previous mentors at MGH, Risa, Keith, and Doreen, who a lot of these ideas they've been such a huge influence for all this. And of course, our funding sources as well. So I will stop there, and I think there are a couple minutes for questions if anybody has them.
Thank you so much, Dr. Mormino, and we'll now move to a few questions we already have. So, Serdar, can you please unmute?
Yeah, I had asked whether the molecular mechanisms are understood with these protein accumulations and Alzheimer's disease, but as far as I understood, it is still an ongoing research, and the details are not understood of these molecular mechanisms.
Yeah, I think it is sorry, I missed. Oh, I see. You did ask a question. Yeah, I mean, I think there's lots of work. Most of that work, would be like in the animal space, like the molecular mechanisms. And I think the question is, how much does this like translate to, the human model? And I think there's a lot of different ideas from the basic science space on the exact mechanisms. I've been thinking a lot about VTC and why VTC might be vulnerable to tau deposition. And there is a literature on neuronal activity and the release of these proteins, as one potential mechanism for why these proteins may be accumulating throughout the lifespan. And I mean, it would kind of make sense with VTC, and we are with visual memory. This is something that is so present, in the human brain, like throughout the lifespan. I mean, I wonder if there is a connection between that specific processing that's necessary for visual information and the release, of these proteins. But I mean, that's I have no data to support that. I just have been wondering why VTC in particular might be so vulnerable to early tau deposition.
So for the animals, at least, do they have concrete results?
I mean, that's just more. So, I mean, there's a variety of approaches, but yeah, the results are very concrete with measuring, they can do the experiment, they can measure activation and then measure, the proteins being released and things like that. But how that translates, to the aggregations that we see in humans and the spatial patterns. I mean, I think that's where the gap probably exists.
Thank you.
Thanks. Next question from Bruce Jenkins.
Oh, hi. Very nice talk. Thank you. So I just had a couple of quick questions. One of them is when you were presenting the tau data, I believe, correct me if I'm wrong, I think, your earliest age you were studying was about 60 years old. Is that correct?
Yeah. So in general,
the data you showed.
In the general, Keith. So I did show one plot from Keith's group, where it does extend earlier. But for the most part, you're absolutely right. The tau data, all the cohorts, is typically 60 and up.
Right. So what I was curious about is when you look at those dot plots and you compare them against the different kind of subtypes, one thing that it looks clear is that as a function of age, it seems like the effect size isn't really that big. which would suggest that maybe you're kind of picking it up right when it really is kind of starting. And I was just, so one thing I was curious about is there enough data to look at earlier time points to figure out if this really is kind of a critical age of inflection, or is there convincing data that it might actually be earlier?
Yeah, I think, the age piece is very complicated because, so on the one hand, when you look at these plots, so like from Keith's paper, using the Framingham data where they start earlier, there's this positive relationship with age, so that like if you look at interrhinal, there's this very subtle increase, when you go from age 40 to 80. However, with these young onset AD cases, these patients, it's the opposite. They like early onset, people who have dementia when they're 60, they have the highest tau levels. So I think there's like two things going on with the age. There's this like aggressive tau production piece that happens in younger subjects. And who knows, there's probably some complex genetics for that. That's probably separate than the age association we expect with interrhinal cortex from like kind of the Brock like literature. So I don't know. I don't think I answered your question. Probably the answer to your question is no, there probably isn't enough data to flush out this aging component. But it seems like people are addressing it from two angles. One is like, how does age influence interrhinal? And we have a lot of reason to believe, like we can just kind of match the postmortem literature for that. There's a ton of data from the postmortem literature on like what we expect with the aging piece with interrhinal. And then I think another approach to this is to go for these atypical early onset patient cases where there's clearly a different process going on with cortical tau in those cases. The thing that was really surprising to me about the A4 data set is that this is just a sampling of older individuals, as you're noting, age 60 and above. They're not selected for an atypical presentation. And still, even in that tight range, 60 to 80, you do see that the people that are closer to 60, if they're amyloid positive, they're more likely to have this cortical presentation. So I don't know, like what, I mean, I guess we just need more data, and we also need to look at these subtle levels within normal controls to tease this apart. But I think the aging piece is twofold. There's two different things going on: aggressive cortical tau in some cases for some unknown reason, and then subtle increases, a subtle older age increase for interrhinal cortex.
And can I ask one quick question? Yes. I don't see anybody else here, but so the other one I had was, now that you're getting, these new tau ligands and you're able to get these nice maps, of kind of other areas where tau is accumulating. Have you compared those to like maps of the hypometabolism by FDG PET, other vascular maps of, regions of lower perfusion?
Yeah, so we haven't done that. Yeah, we have not done that. I mean, there's been a couple papers doing that in the context of patients. We have not been doing that because we're not collecting. I mean, unfortunately, with these normal aging cohorts, what has happened with all these ligands, you kind of pick like two. So it tends to end up being like the amyloid and tau, and we don't do FDG anymore. Yeah, it's a good point. We could look at the perfusion data because we could look at like the first few minutes, of the actual PET scan as like a pseudo perfusion map. And we do have some blood flow measures from the MRI side. But we haven't dug into that from our end to see how redundant it is, or if there is like differences in the spatial kind of contributions across those modalities.
Okay, thank you.
Okay, so since I don't see any questions at this point, I would like to thank you so much again, Dr. Mormino, for coming here today, and hopefully next time we'll see you in person.
Thank you. Thanks for inviting me. Take care.
Thanks. Goodbye, everyone.